Hi, I'm Deka, and me and Yuting will present our final project for ADL, detecting cancer in huge medical images. So we want to develop a tool that assists physicians from training data, and the data source is using Chameleon Challenge, and it has different image magnification levels, and each medical image is called slide. So in this project, we experience how small ML system is compared to the whole environment surrounding it. And we try to reframe our problem to make it easier to walk through it. So this is our pipeline. So we have 5 pipelines basically. So we start with initial data analysis and then data preprocessing, and then we create a model and then feed in the preprocessed data into the model. And then we perform data post-processing, like extracting patches to create a heat map, for example. And then we predict and evaluate our model. So in the first step, we perform initial data analysis. The goal is to understand what the data we have and then plan for the end-to-end pipeline. So we perform lots of stuff like from data inspection, visualization, and sanity checks. We also learn how to use the tools, OpenSlide, Matplotlib, and then we try to understand the medical and technical terms from the paper, like slides, what is patch, what is tumor, and then finding how to handle multiple scale image. And this is what the example of what we found. Like this is the full slide, and then this is the tumor, the healthy, and then this is the tumor in the slide. And then on the end-to-end pipeline, again, we have the 5 steps. And I think it's easier to go through that through a code walkthrough. So we split our implementation into 3 parts. So the first is initial data analysis and pre-processing pipeline, and then the second is the model and evaluation pipeline, and the third is the experiments. So let's just go through the code for a walkthrough. So we have here we have our code for initial data analysis and pre-processing. So first, of course, we run the start code to install things and then import the libraries. And then we'll define several setup, like where to read the slides from, what's the format of the file name, and then what's the model tag, the level that we're doing. Yeah, and then we also during this initial data analysis, we use the image that the profs has provided to play around with the tools to see what the data we have. And then we define our slide related functions. This is also some of it already provided, but we also define our own create tissue mask, which is a binary mask of the tissue, which is 1 or 0, 0 for a tissue, 1 for none. And then here we try to extract an image of slide of level 7 magnification and 4 to compare. And we also learn how to like exactly get the same point of different magnification level, like what is the mapping between them. And then we try to see what the tumor slide has here. So this is all the data analysis, like, and this is the result of the tissue mask that we have. It's a binary image, and then we also define our methods for loading data from Google Drive, and then extracting patches and labels. And this is extremely important because these patches and labels are used for the model pipeline. Yeah. So first we have the is patch tumor. So the task is to detect whether the tumor mask have a tumor in the center. So the center will be defined by 128, 128. So we follow the paper. So the center will be 128 by 128, and then the mask patch will be 299 and 299. And then we'll extract patches. So this is our helpers for extract patches method. So the extract patches will receive slide and tumor mask and the level. So it will first read the full patch and the full tumor. Sorry, it's not patch. It will read the full slide image and the full tumor image, and then it will find the tissue pixels among them, and then create the tissue mask, the binary image that I just mentioned. And then to extract where the tissue is healthy, we subtract the tissue from the tumor image, and then set, if there's any minus value, then we set that to 0. So this will give us all the healthy tissue. And the reason why we separate tissues that are healthy and tumor is because we want to have a balanced data between tumor cells and healthy cells. That's why like we split the two, and then when we extract the data patches, we will count the number of healthy tissues that we get and the tumor cells that we get, and make sure that they're almost equal. And while we're extracting the patches, we're also extracting the data label. So this is where we call the is-patch tumor is called. Like if it's a tumor, then the label will be 1. So we add that into the label, and then we'll save both the slide and the tumor patch. And same for the healthy patch. And it's a healthy patch if the center is not a tumor. That's why we add this condition here to say that it's not a tumor. And then the label for the healthy cells will be 0. Oh yeah, so 1 is cancerous and 0 is non-cancerous. And then that's it for the extract patches. And we also have this function from slide ID. So this will read directly from the Google Drive instead of reading the slide one by one. So we can just pass in the slide ID, and it will automatically load the file from Google Drive and extract patches. And we also perform some optimization. So if we already pre-processed the file before, then we'll just reload instead of extracting patches again. So the first time we extract the patches, we will write the patches and mask and labels. But if it's already there, we'll just reload them unless the force reload is true. So that's how we extract patches. And then we also want to perform some sanity checks to check whether the extracted patches and masks are really, really correct. So here we define a simple function to like randomly plot patches and mask, and this is what we got. This is what we got. So when we sample the patches and plot it, as you can see here, like for label 0, meaning the cell is healthy, we don't see any cancerous cells in the middle of the patch. But if you see the ones that labeled 1, then the center of the patch will have some tumor. So yeah, we did this a lot of times, and we believe that it's correct. The implementation for extracting patches is correct. So yeah. And then the last part, because we want to optimize the pre-processing part, we pre-compute all the several slide images at once. So we extract the patches from multiple images at once and save that to Google Drive for further subsequent processing, for example, during training and modeling evaluation. So that's part 1, initial data analysis and pre-processing. So next, we have this second pipeline, which is the modeling and evaluation. So again, we install the required libraries and import the required libraries, and we have the relevant slide functions, and this patch extraction is the same as before. The difference is now, instead of performing patch extraction, we'll actually just reload what we have pre-computed before. So here we just we have this utility to read from several slide IDs and return like what are the train patches and the test patches. So we declare where to find these models and then some constant which define our experiment. And then we also split the slide IDs into train and test. That's why when we call that function, we'll get like a separate set of training and test. And then next, we'll call this train test split to split the train patches into training and validation sets. And that's it for loading data and splitting data into two sets. And then here, during the modeling part, we'll define a model. So here we support two models. We're using transfer learning. We can either use VGG or Inception. And then here, during the training, before we do the training, we will rescale the data by 255. So both training and testing will receive the same operation, which is rescaling the image by 255. So we're using the data generator from TensorFlow. And then here we have the load latest checkpoint and load latest model, which basically just reload the latest model, the best latest model that we have available for a certain model tag. So model tag is like the model ID. Like if we have different models, then they'll have different model tags. And then here we will train our model. We define a function for that, which will receive train data gen and fail data gen. And then this will basically train the model. And then we also declare like two checkpoints, like early stopping. So it will stop if after 10 subsequent steps there's no improvement, then we'll stop the training. And then we also perform lots of model checkpointings. So if the model is found to be one of the best performing among all, we will save that model. Yeah, and then at the end we'll save the model to drive. This is necessary because in Colab it's sometimes not stable, so it's best to save as much as we can. And then we also declare a function for plotting the accuracy and validation of the training. Yeah, and then here during the model training, you can either load an existing model, which will just reload the best latest model, or you can create from scratch and train it. So the epoch is default to 50, and you can pass in the model tag for your model. And you can select model base that you want, either VGG or Inception. So this is the plot of the training and validation accuracy. And then next we'll have the model prediction. So before we do prediction, we must do data post-processing. So for any image that we want to predict, then we need to extract patches from that image. So we'll use again the read slide and function to get the slide patch and the tumor patch. And then we'll just predict if that patch is found to be a tissue. And then we'll extract what's the label. So we can use the is-patch tumor that we have explained before to check whether the centroid of that patch is a tumor. So from this function, we'll get three things. First is the coordinates of the patch, and then the test patches and the true labels. And then during the predict test data, then we'll receive that test patches, and then the model will predict the test patch for that test patch. And during the evaluation method, we perform several scorings, and Yuting will explain them, why we choose them later. And then we have this plot probability heat map. So it will read the full slide and full tumor. And then we also declare the heat map overlay, which will plot the probability heat map. So the way we do that is we'll set the value of the heat map overlay to be the probability, and then we'll add like the opacity to the colors. If they are very high, then it will have a higher opacity, but if they're very low, then they'll have a lower opacity, like very transparent. And then we have evaluate model on slide, which combines everything from reading a slide and then extracting the test patches and the true labels, and then predicting the test patches, evaluating them, and plotting. So this is the example of how we do that. Like we pass in the slide ID, and then it will just do a prediction on that image, like what are the evaluation metrics, and then the heat map. So that's it for the walkthrough of the code, I guess. Yeah, so as I mentioned, we did this in the walkthrough. And then during the patch extraction, we are using 299 by 299 patches with 128, 128 center. And if the label is 1, that means the centroid contains a tumor. But if it's 0, then it's healthy, like the centroid doesn't contain a tumor. And we also handle data imbalance by sampling tumor and healthy tissue pixels with almost equal probability. And as for data loading and saving, we save lots of things to Google Drive.
So here comes the modeling and evaluation part. We try to use transfer learning because we want to reuse existing model instead of starting from scratch. And we use convolutional based both inception model and VGG. And our implementation includes first data loading, and we split the data training and validation, and then we create models, and then we training, and after that we post processing, and we predict the data and evaluation, and then we test and evaluate. For the evaluation part, because the tumor detection is very imbalanced, so we are now using accuracy, and because it's not the best metrics to use, so we are going to use the precision and recall to count how many positives are actually positives. And also we use ROC and AUC score to avoid overfitting into one of the class. And for the result, the first one, it shows the heat map with the average precision score 0.62, and there are some of the false positives because the training set is very limited. And the second one, the precision score is a little bit higher. And the third one, there's a very low precision scores. There are too much false positives because of the limited training slides. And we also use the recall score to evaluate the heat map too. And for the experimental results, we
did 2 experiments. One is for the model, and the second is with the slide level.
Yeah.
Okay.
We compare the VGG and inception. The VGG performs better than inception, because it has a higher precision score, and the inception model doesn't perform very well. There are too much false positives for level 4. And we also compare level 4 and level 5. As we can see, the level 4 has a significant higher precision score, 0.75, compared to the level 5. That's why we are using the VGG model
for the base model for everything.
Yeah.
Okay.
Thank
That's
you.
all. Thank you.